That's pretty decent.
This doesn't sound right.
It's just a piece of chicken rolled up as the shell.
I never tried that example. Anytime I try to go to, you don't have,
They have everything and they put
and
it
it only starts hours, and then I use it.
That's not that cheap.
It's like that KFC sandwich they
Yeah.
That's only healthier.
No, where's the bread at? I'm not eating this.
You're welcome.
What do you find? Where's that fish? I eat this.
All right, guys, we ready to get going? So today we're going to talk about Chapter 7, and this deals with microbial nutrition, ecology, and growth, also known as a really dry chapter. Okay. Really, all this chapter deals with is how microorganisms utilize nutrients from the environment to then grow. So then we'll talk about how they actually grow, and then also how they interact with each other in the environment. Okay. So we'll start off with nutrients, and nutrients are essentially sources of elements for the cell. Okay. Cells need to acquire elements. Okay. Without these, they would not be able to build things within them. Okay. The most common nutrients are going to be the top 6 in this list: carbon, oxygen, nitrogen, hydrogen, phosphorus, and sulfur. These are the most common, and these are the ones that are required, usually in fairly high levels. Okay? Everything below that, potassium, sodium, calcium, magnesium, chloride, iron, manganese, blah, 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 blah. These are what we call trace elements, or trace nutrients, and they are required by most cells, okay, but in small amounts. Okay? And they usually serve very specific functions, usually activating enzymes. So we won't focus on those guys too much. Instead, we're going to talk primarily about the top 6. So what is a nutrient? This is anything a living organism needs to survive. Okay? It needs to acquire it from the environment. And again, we usually reference this as its basic elemental composition. Okay? So this organism needs this much carbon atoms. Now, obviously, it's not going to be just picking carbon atoms out from the environment. Instead, it's going to get those carbon atoms stuck to other atoms, and this is what we call a molecule. Okay? So most of these, we're talking about their elemental form, but most of the time it's coming in a more complex molecule. Okay? An essential nutrient is anything that must be provided to the cell. Okay, that's what essential means. If it's not there, the cell dies. And we usually categorize these into 2 groups: inorganic and organic. Inorganic is basically anything on the periodic chart. There are variants of it. Organic are compounds or molecules that have calcium, carbon and hydrogen. Okay, so carbons and hydrogens, if that's in a molecule, it's considered organic. It can have any other kind of stuff stuck to it, any other elements, but carbon and hydrogen are the bare requirements. Carbon by itself, or hydrogen by itself, or carbon with something else other than hydrogen, is not considered organic. Okay, it needs to have carbon and hydrogen. Most of the molecules that living cells require are going to be organic in nature. Okay. Now, prokaryotes can be a little iffy, especially archaea. They can actually live off of inorganic molecules, but most living things require organic nutrients. So here's an example of E. coli, and its different compositions of these materials. Okay? So you can see carbon at the top of the list makes up 50% of the dry weight. We talk about dry weight because in reality a cell is 70-80% water. Okay? And we don't want to talk about that just because we know that's there. Instead, if we dry everything out, carbon makes up the vast majority. And this makes sense because all organic molecules use carbon as a backbone. Right? That's what an organic molecule is by definition. Next, we have oxygen at 20%, nitrogen at 14%, hydrogen at 8%, phosphorus at 3%, and sulfur at 1%. Okay? Those are the 6 big ones that we're going to talk about in more detail. All these other ones you can see are found 1% or below. Again, these are trace elements, and again, they're used primarily for specific enzymatic functions. So I'm not going to say they're not important, but they aren't as important as all the others. So the other top 6. So let's talk about where some of these essential nutrients come from. So we'll start off with carbon. This is the most abundant, as we mentioned before. It makes up 50% of the cell. And for most organisms, at least the ones that we're going to focus on, they're going to be considered what we call heterotrophs. And this essentially means they get their carbon from organic molecules, okay? And their organic molecules are literally stolen from usually another either living or dead thing, okay? The most common organic molecule that is used for a carbon source is glucose. Why? Because glucose is one of the most simple organic compounds that are made from inorganic compounds, okay? So heterotrophs, these are guys who get their carbon sources from organic molecules, and they take these from other living things, okay? In the case of parasites, these living things are going to be actually alive, okay, like us. In the case of saprobes, or organisms that like dead decaying material, it would be a once living thing, okay? It is now dead. Autotrophs, on the other hand, these are organisms that produce their own organic molecules from inorganic molecules. As I mentioned before, glucose is the most common organic molecule used by cells, and glucose is actually produced by autotrophs. Autotrophs can take carbon dioxide, which is inorganic, water, which is inorganic, and a little bit of sunlight, and produce an organic glucose molecule from it, okay? Nitrogen, so where do we get our nitrogen from? Well, nitrogen is very, very abundant. It makes up about 70% of our atmosphere, okay? However, most organisms cannot directly take in nitrogen from the atmosphere, okay? Prokaryotes, on the other hand, there are a large number of prokaryotes that can do this, and we call these guys nitrogen fixing, okay? So a nitrogen fixing organism takes nitrogen from the atmosphere and fixes it usually into an ammonium ion, okay? This can then be utilized by other organisms that can't do this for its nitrogen source, okay? So it ultimately comes from the atmosphere, but it's only able to be transferred from the atmosphere to other molecules by very specific prokaryotic organisms or bacteria. Oxygen. So oxygen is 20% of what we breathe, and unlike nitrogen, all organisms that utilize oxygen can take it directly from the atmosphere. Okay? This one's easy. You have your lungs, you have a membrane or whatever that allows the oxygen to diffuse into the body. Hydrogen. So hydrogen is another relatively abundant molecule in cells. And this makes sense because, as we mentioned before, for a molecule to be organic, it needs to have a carbon and a hydrogen. Most molecules in cells are going to be organic in nature, so you would expect to see a lot of these hydrogens. Now, not only do they make up organic molecules, they can also be used for buffering or maintaining a pH within the cell so that it doesn't become too acidic or too alkaline. These hydrogens are also really, really important in forming hydrogen bonds, and hydrogen bonds are weak attractions of molecules. And we'll talk more about what that means in Chapter 9 as far as the cell is concerned, but it's important for things like DNA and RNA. And last but not least, hydrogen ions can actually be utilized to store energy. And this is how cells store energy in the form of ATP, which we'll talk about in Chapter 8. Phosphorus, it's often found in a form called phosphate. This is a naturally occurring compound that is found literally everywhere, like a mineral, and it's usually brought into the system by plants, which then other organisms can steal later on. Okay? Again, this is a relatively important molecule for cells, not only because it's used to produce things like DNA and RNA, but also in the production of ATP. ATP stands for adenosine triphosphate, so those P's are literally these molecules. So it's important that they're present. And then lastly, sulfur. So sulfur is kind of on the cusp of being a trace element, but we don't really consider it one because it's really essential in the production of proteins. Okay? So a lot of proteins use sulfur to create disulfide bonds, which is basically a way for things to stick together. So without sulfur, you wouldn't get complete proteins, and then the cell would die. So this is why it's considered a little bit more important than trace. And then these are all the trace nutrients, as I mentioned before, and they all have relatively specific functions. They're not kind of broad like all the other ones that we just talked about. So we won't spend much detail on them. Alright, so those are the essential elements. Now let's talk about the essential molecules, okay? So an element is going to be literally what's on the element table. Carbon, oxygen, nitrogen, phosphate, those things. Where an essential growth factor is a whole molecule. So a bunch of those atoms stuck together, okay? And an essential growth factor is essentially an organic molecule that needs to be produced or needs to be presented to the cell. The cell cannot produce it itself. So this essentially means it's a complex organic molecule that needs to be taken from the environment. Okay? If it does not do this, it will die. Now, you can assume that cells can make a lot of organic molecules, which they can, but there are some examples of organic molecules where cells just cannot produce these. Okay? An example in humans would be the essential amino acids. The essential amino acids are the amino acids that our body cannot produce on its own. So instead, we have to steal it from somewhere else. Either from an animal, a plant, or some sort of microorganism. Now, as we mentioned, carbon sources, these can come from either autotrophs or heterotrophs. If you're an autotroph, you get it from inorganic molecules like carbon dioxide. If you're a heterotroph, you're getting it from organic molecules that you take from other organisms. Okay? Conversely, we also have to talk about where does the energy come from. So energy is used by the cell to make things. And we'll talk all about how this occurs in Chapter 8. But without energy, the cell will die. Okay, so it's an important aspect of the cell's life. By far and large, most organisms are what we consider chemotrophs. And chemotrophs use organic molecules, okay, to produce energy, okay, store energy. Again, the most common organic molecule used is glucose. But really, most organic molecules can be processed, broken down, and their energy stored. Now, there are some organisms that can use light to produce energy, as well as lithotrophs, which can use organic molecules. But neither of these are going to be pathogenic, so they're not going to be of interest to us. Okay? We're interested in the heterotrophs because they have to steal organic molecules from outside or from other organisms, and chemotrophs, which then use these organic molecules to make their energy to survive. Now, for microorganisms, when we look at chemotrophs, there's two categories. Saprobes and parasites. Saprobes, by far and larger, is where you're going to find most microorganisms. These are guys that like dead material. So they get their organic molecules from once living things. Okay? So a dead tree or a dead plant or a dead body. They are not getting it from something that is alive. How do microorganisms get these nutrients inside of them? Well, they release something called an enzyme. And enzymes are proteins that can serve as catalysts. And essentially what that means for this portion of the talk is when they are released, they can break stuff down. Okay? So they're released by the bacteria. They break down these large, dead things into small little baby organic molecules, which then easily can be taken up by the cell. So they literally digest from the outside and then bring it in. The other group of chemoheterotrophs are guys who use organic molecules for energy, are parasites. And these are the ones that we are going to be most interested in because these are the ones that cause disease in humans and other organisms. Okay? These guys like to get their carbon sources from living things, okay? Not already dead. And we often call these pathogens because when they do this, you get tissue damage and destruction. Okay? When they come in, they take over, and they try to utilize your organic molecules. In the process, they destroy tissues, and this makes you feel sick. Now, these organisms can be on the outside of the body, which we call ectoparasites, on the inside of the body, which are known as endoparasites. Within cells, this is known as an intracellular parasite, or an obligate parasite. And an obligate parasite is often called a true pathogen. And that means it has to live off of another living thing or it will die. It cannot live on its own. It's dependent on a living organism. Alright, so those are the nutrients. Now let's talk about how the cell actually gets these nutrients inside. Okay? So hopefully you remember from chapter 4, we briefly talked about the cell membrane. And I said that it was a phospholipid bilayer, which looks something like this. You have phospholipids, which have a phosphate head, which is the round part, and then a lipid tail. And there's a bilayer, which means there's two of these. Okay? One on the outside and one on the inside of the cell. And when these connect all the way around the cell, like so, you get your membrane that completely encapsulates the cell. Okay? Now, hopefully you remember when we talked about this, I said that these guys are hydrophobic. Okay? Which ultimately means they have a charge associated with them. Okay? The problem that we're having here, when you want to get nutrients in, is that oftentimes nutrients have a charge as well. So what happens? Like charges repel. So if this cell wants to get this nutrient in, nope, it's going to bounce right off. Okay? So how does the cell get around this? How does it actually let in nutrients? Well, it does so by producing something called a carrier. And a carrier is basically a protein pore, or hole in the cell. And this pore will have the opposite charge of the molecule that you want to let in. So now instead of this being repelled, it goes right into the cell. Okay? Why does the cell go through all of this trouble? Well, it does this because it gives it great control over what gets in and what gets out of the cell. If the cell wants this molecule in, it puts this pore in, boom, bing, bang, boom, it comes in, it's got it. If the cell decides, I don't want this anymore, gets rid of this guy, membrane repairs itself, now it's not coming in. So this acts kind of like a gate, okay? This allows things to leave or enter the cell as the cell sees fit. Now what we just described is an example of passive transport. And that's where things just naturally come into the cell without any energy. Okay? This can only occur if what you're trying to get in or collect is more abundant on the outside of the cell than on the inside. Okay? If for whatever reason this isn't the case, and instead you've got more of your stuff on the inside than the outside, you cannot go through this same process. The reason for that is if we follow the laws of natural diffusion, the molecules would then go this way instead of the way we want. So in that case, you need to utilize energy. And energy comes in the form of ATP. You hydrolyze this, bing, bang, boom, you can bring things in against the gradient. Okay? So, getting nutrients into the cell is complicated. Okay? It is not something that is easily done by the cell. It requires energy and it requires a lot of stuff for it to work properly. But it does this because if it doesn't, then it will die very, very quickly. Because then it's no longer in control of its environment. So, before we get into all the nitty-gritty, let's actually talk about some basics. So first and foremost, diffusion. Diffusion is a basic law in science. It basically states if you take something with a concentrated molecule, like let's say a sugar cube, okay, so a whole bunch of sucrose molecules, and you put it into a liquid that has no sucrose molecules in it. Over time, without doing anything, so no energy needed, no stirring, nothing magic, these sucrose molecules will leave from their concentrated source and spread out throughout that liquid until they are equally spaced apart. Okay? This happens naturally. This is a natural occurring phenomenon. No energy is required. Okay? And this happens with everything. Concentrated molecules will go from high concentration to low concentration until everyone is equal. Okay? Now, when we talk about any molecule but water, we call this diffusion. When we're talking about water in particular, we call this osmosis. Why the difference? Well, it all has to do with the cell membrane. So remember before I said the cell membrane is hydrophobic and hydrophilic in the middle, and that most things are charged and will repel away? That's true with most things, but not with water. Water is non-polar, which means water can relatively easily wiggle its way through the cell. What that means is the cell doesn't have much control over it. Okay, so it's got to come up with clever ways of preventing these water molecules from doing damage. Okay? So let's talk a little bit more about what this actually means for the cell. So in this experiment we have on the board, we have what we call a dialysis membrane. A dialysis membrane is basically a piece of saran wrap with really tiny holes in it. Okay? These tiny holes are large enough so that water molecules can easily go through it. But larger molecules like a salt or a sugar cannot. So this is very, very similar to a cell membrane. Okay? Water molecules can wiggle through, no problem. But bigger molecules like a solute, which is just another word for salt or anything dissolved in a liquid, cannot. Okay? So let's take this dialysis membrane and form a sac. So it looks like a cell. Completely covered on all sides, just like a cell would be. And then inside of this, let's put some salty water. Why salty water? Because that's what cells look like. Now let's take this and then stick it in a beaker with pure water in it. Anyone want to guess what would happen?
The membrane expand because all the water is going through, so there's no parts.
So all the water would be going through. Why would the water want to go into the cell? Think about diffusion. Remember, diffusion is where all molecules want to be equally spaced in the environment. So what's happening is water is going to rush in because it's going to try to make the same amount of salt on the inside as there is on the outside of the cell. Is this possible? Can the salt get through the membrane? No. So the salt will never go into the beaker. So it's never going to equal out. So the water molecules are just going to keep going in and in and in, trying to equal out the amount of salt on both sides, but it's never going to happen. So what does happen? It explodes. Now everything's equal, because the membrane is gone. Okay? Now let's flip this around. What if I have a cell that's just pure water, and I stick it in a beaker with a whole bunch of salty water? What would happen? The water in the cell leaves because it's trying to equal out the amount of salt on the outside as on the inside. Is that possible? No, because the salt can't leave through the membrane. So instead the cell shrivels up into a little turd. Okay? So, why are we talking about this? Well, as I said, the cell doesn't have much control over water. It goes in and out whenever it wants. So if the cell all of a sudden goes into like a freshwater lake where there's no salt, it's got to worry. It's got to figure out a way of preventing the water from rushing in and causing it to explode. And then if it swims into something that's got a lot of salt, now it's got to worry about not shriveling up and dying. Okay? When we talk about osmosis, we usually talk about it in context of where the organism is. Okay? What does the environment look like? How much salt is in the environment compared to the cell itself? So we break this up into three categories: isotonic, hypotonic, and hypertonic. Isotonic is where you have the same amount of salt on the outside of the cell as on the inside. This is the best case scenario for the cell. Because in this case, the salt is equal. So diffusion is already set. Everyone's happy. So the water molecules go in and out at the same rate. The cell's got nothing to worry about. Hypotonic is where you take the cell and you put it into a solution that has hardly any salt in it. Okay? In this example, water is going to rush in, trying to equal out the amount of salt on either side. That's not possible, and the cell will explode if it does nothing. However, there are ways that cells prevent this. Okay? For bacteria, they have a cell wall. Their hard, rigid cell wall creates a thick, strong structure, so when the water comes in, the pressure from the water doesn't cause it to explode. For animal cells, it's a little bit more difficult because they don't have a cell wall, just a membrane. So what they'll do is they'll actually collect the water in a little hole or vacuole and then spit it out. Okay, so it doesn't cause it to explode. Lastly, a hypertonic solution. Hypertonic solution is where you have more salt on the outside than on the inside. In this scenario, if the cell does nothing, all the water will rush out of the cell and the cell shrivels up and dies. Again, bacteria use cell walls to prevent this from happening. And for animal cells, it's very, very, very difficult, so there aren't many animals that live in this situation. So here's just a cartoon diagram of what this looks like. So isotonic, same amount of salt on either side, the cell membrane, water goes in and out freely, cells happy. An example of an isotonic solution in nature is the ocean. The ocean has the same amount of salt in it as most organisms, okay, including us. Hypotonic solution, with more water on the outside than on the inside, and so the water rushes in, trying to equal itself out, the salt, and when this happens, the cell will explode if it does nothing. Example of a hypotonic solution would be a freshwater lake like Lake Erie. And then a hypertonic solution is again where you have more solute on the outside than on the inside. Water leaves the cell, it shrivels up and dies, it does nothing. And an example of this would be, let's say, the Great Salt Lake, right? A really saline, salty environment. Again, freshwater organisms. If you're a bacteria, your cell wall protects it from bursting. If you're an animal cell like an amoeba, you have special vacuoles that collect and push the water out. Okay? There is a reason why there are way more organisms in the ocean than there are in the fresh water. Not only because the ocean is much larger, but because it's easier to live in an ocean. Because the salt's the same as inside of the cells of most of those organisms. High salt conditions, as I said, and these are hypertonic. The Great Salt Lake is a prime example. Most organisms cannot live in this type of condition. Okay, so if you go to the Great Salt Lake, you'll see there's hardly anything there. However, there are some very specialized organisms, primarily heliobacteria or archaea, that can live in these really salty, nasty environments. Most high-order organisms cannot, and that's because salts interfere with neurological transmissions. So you can't really be a high-ordered organism with all the salt around. It doesn't work very well. Okay, so that's water. What about everything else? So we've already kind of talked about this, and we said that when the cell wants something to come into it, it creates a hole called a porin. It's known as a protein carrier as well. And this allows the molecule to enter the cell through the membrane. Okay? Now, if the cell wants to do this without utilizing any of its energy, without wasting energy, it can do that in a process known as facilitated diffusion. And all facilitated diffusion is exactly what we talked about, where you have a molecule trying to get into the cell, and it goes through a pore without needing any energy. But the only way that this is possible is if the molecule is going with the concentration gradient. And what I mean by that is, there's a higher concentration of the molecule on the outside than there is on the inside. When this is true, natural diffusion will occur without any energy, because again, you're trying to get the same amount of these guys on the outside as on the inside. Okay? Unfortunately, that's usually not the case. Most of the time, you're trying to go after something that is scarce. Okay? It's something that you're trying to collect from the environment. So in that case, you're going against the concentration gradient. So you're going against the concentration gradient. The molecule that you want to collect, let's say glucose, I'll write that as a G. You've got more of it on the inside, and there's only a little bit of it on the outside. If you were to let diffusion take over, do it naturally, it would go against what you want. These guys would leave, right? Because it's trying to equal out on both sides. That's a no-no. So what does the cell do instead? It says, nope, I'm not going to let that happen. Instead, I'm going to give ATP or energy to my protein carrier, and now it can literally grab onto the glucose and bring it into the cell against the concentration gradient. And again, this only occurs or only happens when the concentration gradient is the opposite of what you want. So if you're collecting something, that means you have more of it on the inside than on the outside. Again, this is by far and large more common, so that what that means is the cell constantly needs energy in the form of ATP to bring in these scarce molecules. If you are an animal cell, you've got a couple other tricks up your sleeve, and that's because you do not have a cell wall. So that means you can move around your fluid. So if you see something you really like, you don't have to break it apart into little pieces and then slowly let it get through your membrane. Instead, you can just go up to it and cover it up and engulf it. Okay? This is a process known as endocytosis. Alternatively, if you have crap inside of you that you need to get rid of, you don't have to break it up and shoot it out piece by piece. You can just push it out, and this is called exocytosis. Okay? So endocytosis, bringing particles in, exocytosis, bringing particles out. Some specific examples of endocytosis, so phagocytosis. This is where you have special cells in your body called phagocytes that literally endocytose foreign things, including bacteria. So they come up to a bacteria, they go, you're bad, I don't like you, and they just gobble it up in one go. Penocytosis is endocytosis but with a liquid. In this case, the cell creates a little, almost straw-like apparatus that sticks into the liquid and just sucks it into the cell. Okay, so those are nutrients and how the nutrients get into the cell. Now let's talk about some of the other things required by the cell to survive, okay, other than nutrients. First and foremost, we have temperature. And with all organisms, there is a minimum, a maximum, and an optimum temperature. Minimum temperature is the lowest temperature the organism can grow. Maximum temperature is the highest temperature the organism can grow. And optimum temperature is its favorite temperature, the one where it grows the best. With bacteria, we have a wide variety of organisms that can grow at pretty much any temperature on Earth. Okay? This is a lot different than what we would see with higher order organisms like ourselves. So what we do is we break them up or categorize them based on the temperatures that they can grow at. Okay? So we'll start off with the blue guys, which are the Saprobes, and they can grow anywhere from -15 all the way up to about 4 degrees Celsius, which is basically your fridge and freezer. Fridge is 4 degrees, your freezer is about -20 or so. And their optimal temperature is, right around 2 to 3 degrees. Which means they like to grow at very, very cold conditions. These guys are obviously not going to be pathogenic. Our body is 37 degrees. If we eat one of these guys, it's going to die. Okay? Next, we have the Psychotrophs. It's the yellow guy. Goes anywhere from 5 degrees all the way up to 35 degrees, with an optimal temperature of around, let's say, 23 degrees. 23 degrees is room temperature. So this is where you're going to find most environmental organisms in this category. Next, we have the Mesophiles. Mesophiles are anywhere from 10 all the way up to 50, with an optimal temperature of around 37 degrees. These are the guys that are going to be either potential pathogens or organisms that live within us. Why? Because our body is 37 degrees, okay, on average. So that means these are the guys that we're going to be most concerned with, the Mesophiles. Next, the Thermophiles, 45 up to 80 with an optimal of around 70. And the extreme Thermophiles, 70 up to 130, an optimal of around 125. 100 degrees Celsius is boiling water. Okay, so these guys live in really, really nasty, hot conditions. Okay, so you can see bacteria grow in all different varieties of temperature. But again, the ones of most interest to us are going to be these mesophiles. So psychrophiles grow in really cold conditions. Most of these guys are going to be found in the poles, so Arctic or Antarctic. Here is an example of what they look like. You could probably barely see it. This brown crap in the snow isn't dirt. This is the organism. Okay? So this just shows you that they're very, very simple. They can't be that complex at those extreme temperatures. They're literally just these little photosynthetic blobs. Mesophiles, these are organisms that grow at around 37 degrees is their optimal temperature. So again, these are going to be the ones that are going to either cause disease in humans or live among us. Thermophiles, these are the guys that grow at really high temperatures, in some cases above boiling. These guys you'll find in very extreme conditions. So maybe, at the Marianas Trench, the very bottom of the ocean where there's lava being spewed out at a constant rate. Again, not something you would find a pathogen. All right, so enough about temperature. The next environmental factor that is required for growth are gases. And when we talk about gases, we usually talk about oxygen and carbon dioxide. When we're dealing with heterotrophs, oxygen is required and carbon dioxide is a waste product. Okay? When you're dealing with autotrophs, carbon dioxide is required and oxygen is a waste product. We're mostly going to be dealing with heterotrophs because those are the guys that can cause disease in humans. And so we're going to focus on oxygen because oxygen is required and so they need it to survive, at least in most cases. Now when you have bacteria, you can actually have bacteria that cannot utilize oxygen. Okay, at least elemental oxygen, or the stuff that we breathe. In that case, we call those organisms anaerobic. Okay? However, there are bacteria, in particular, that can also utilize oxygen. In that case, we call them aerobic. So what we end up with is a continuum. And by continuum, I mean you have aerobic organisms on one side, and these like O2. And then on the other side, you have anaerobic organisms, which hate O2. And if we were to make this a line, there's going to be a couple different organisms, or different classifications of organism, based on these oxygen requirements. If you have an organism that absolutely requires oxygen, we call this an obligate aerobe. And that would be this guy here. Okay, this is the obligate aerobe. Next, you can have an organism that we call a facultative anaerobe. And that guy would be right around here. Oh, sorry. Facultative anaerobes don't really care. They can utilize oxygen, or they cannot. They don't really care. They can go either way. These guys are usually the most common, especially within a human. And that's because when they're inside of our body, there's very little oxygen available. So an example of a facultative anaerobe is E. coli. It lives in our gut most of the time where there's very little oxygen. So in that case, it is anaerobic. It doesn't use oxygen. However, if I were to pull that E. coli out, put it on a petri dish and stick it on the table, it would start using oxygen because it's present. So they can flip-flop and go either way. Next, we have microaerophiles. I need to get a better marker in here. Microaerophiles need oxygen, but they need less oxygen than the aerobes. So this is less oxygen, so less than the 20% that we see in the atmosphere. So these guys usually live in water. And they live far enough below the surface where there's less dissolved oxygen. Anaerobes. So these guys do not use oxygen. And if they are a obligate or strict anaerobe, that means if oxygen is present, they die. Or they become an endospore. So an example of an organism that is like this is Clostridium organisms. All of them are anaerobes. So when we find them in the environment, they are endospores. Why do we care about these guys? Because they are human pathogens. Okay? So just because they don't like oxygen does not mean they can't cause disease. A lot of people ask me, where did they come from? How did they even grow if because there's oxygen everywhere? Well, the answer is they were around probably millions of years ago when there was much less oxygen in the environment. So that endospore that's in the soil for tetanus or botulism is probably millions and millions of years old. Lastly, we have aerotolerant anaerobes. These guys would be over here. Just as their name implies, they can survive in the presence of oxygen, but they do not use it. Oxygen. So if oxygen's there, they're cool, they don't care, but they do not utilize it for anything important. Okay. Carbon dioxide. So as I said, in heterotrophs, this is going to be a waste product. So we don't really care about it because it's just excreted out of the organism. So enough about gas. pH is also an important factor. Most of the organisms we're going to be interested in are what we call Neutrophils. These are organisms that grow at around a pH of 7, or neutral conditions. Why are these the ones we care about? Because that's what our body is. It's at 7.4, neutral condition. Okay? However, there are microorganisms that can live in acidic conditions. These are called acetophiles, or in alkali or basic conditions, and these are known as alkalophiles. Excuse me. Acetophiles are by far more common than alkalophiles. In fact, you can find organisms that grow anywhere from a 0 to 1 pH. 0 to 1 pH is very, very, very acidic. It's like battery acid. You stick your hand in there, you'd have no flesh or bone left. Okay? However, it's relatively rare to find organisms that live in very basic conditions. Okay? Generally speaking, basic conditions are very caustic, nasty things, and so it's very, very difficult to find organisms that can live in anything above a pH of 10. And for this reason, most of our chemicals we use to break down or kill microorganisms are basic in nature. Okay? Because it's much more potent at destroying the organism. Osmotic pressure. So osmotic pressure is the amount of water around the organism, and conversely the amount of solutes or salts in that environment as well. So generally when we talk about osmotic pressure, we talk about how much salt is present. Most organisms like to have the same amount of salt on the outside as on the inside. However, there are organisms that like really high salty conditions, and these are known as halophiles. It can use up to 25% NCl, which is really high. And then there are many, many other factors, most of which, we can talk days about. So I'm not going to talk about any of them in particular. Okay? We talked about the big ones. All right, so that's enough about the requirements for organisms to grow, okay, or survive. Nutrients, temperature, pH, and gases. Now let's talk about how organisms interact with each other in the same environment, okay, or in a shared environment. This is a concept known as ecology. Ecology is just how organisms interact with each other. Okay? We'll first talk about symbiosis. And symbiosis is where you have organisms that live in the same environment, and it's required or obligatory. Which means one of them at least requires the other one present. If it's not there, it dies. Okay? This isn't as common, okay, as the other group that we're going to talk about later on, but it does still exist. Within symbiosis, you've got a couple different groups. Mutualism, this is where both organisms need to live in the same environment and they both like it. They're both happy about it. This is extremely rare. Okay? Most of the time, organisms don't want to be around each other, okay? Because they're all going after the same scarce resource. An example of mutualism, so for bacteria, it would be producing a biofilm. A biofilm, you also often have multiple species of bacteria in that same biofilm. They all want to live there because they get protection from the film, but they're all different species. They're all happy about it. For bigger organisms, an example would be a clownfish and an anemone. Okay? Clownfish lives in an anemone which has stingers and protects it. An anemone gets food from clownfish. They both happy. Commensalism. Commensalism is where one organism benefits and the other one could give two shits. Okay? An example of commensalism are most of the organisms in our body. Most of the organisms in our gut are called commensals because they're there, taking up space, getting food, getting a warm space to live, and we don't really care. It doesn't bother us. Okay? Satellitism is where one organism produces something and releases it, and another organism comes along and uses that as food. And it's required for that organism to get it. So it's kind of like mutualism or commensalism, except the organisms are not interacting directly. It's an indirect relationship. And parasitism, this is where one organism benefits and the other one is detrimented or hurt by this relationship. So again, this is where we would find parasites, pathogens, okay? These are where most of the organisms we're going to talk about are going to be found. Here's an example of satellitism. So up here we have Staphylococcus aureus. It's growing on this agar. It's releasing something, some kind of crack, and that's diffusing through the membrane. And now this homophilous organism can grow because it's getting that nutrient that it needs from this S. aureus. But note that they're not growing on top of each other. They're not interacting directly. They're just taking something that one is producing and using it. The other category of ecological relationships are non-symbiotic relationships. These by far and large are the most common we see in nature. Okay? This is where organisms live in the same environment and they don't really care who's there. Okay? Ideally, organisms would want to be by themselves, but there's not enough space, so organisms have to live together. Okay? Within this, there are two groups, synergism and antagonism. Synergism is where these organisms are living together in a shared environment. It's not required, but they like it. They're happy about it. Again, relatively rare. We don't see this too often in nature. Antagonism is by far the most common out of everything we've just talked about. Antagonism is where all the organisms live in a shared environment. It's not required, and they all hate each other. They're all trying to kill each other. They're all trying to go after the same thing. This is life, right? And this is exactly why it's the most common. Because this is the best way to survive. Fighting for everything you can get. So as I said, biofilms. This is an example of synergism, where you have organisms growing in a shared environment. They like it. They're happy about it. But again, this is relatively rare in nature. How do organisms interact with humans? So as I said, there are a lot of organisms inside or on humans at any given time. And these are what we call the normal microbiota. Okay? Most of these are going to be commensals, which means they're in there taking up space, eating some of our food, living in a nice warm body, but they're not really doing any harm to us. Okay? And we don't really care that they're there. Some of these will be beneficial. Maybe they're in there and they're producing a vitamin, like vitamin B12, something we can utilize. And then some of these can be parasitic. And that means they get in, they cause disease, they make us feel sick so that they get more nutrients. They're benefited and we are hurt by it. Okay, so that's ecology and how organisms interact with each other. Now let's talk about how organisms grow. So what happens when they've got all the nutrients they need and they've got all the space in their ecological niche? How do they then reproduce? Well, there's two ways that an organism can grow. It's much like a human. It can either get fatter or it can make babies. Okay? So usually the first thing that happens is the cell gets fatter. Once it gets too fat, it says, okay, I've had enough of this. Now I'm going to divide into another cell. Okay? When a cell divides and it's a prokaryote, bacteria, we call this process binary fission. One cell literally becomes two identical cells. So this is an example of binary fission. You have one cell, duplicates its chromosome, it then splits down the middle and creates two new identical cells. I say identical with quotes because they are supposed to be identical, but if any errors develop during the replication of this chromosome, you'll get mutations, and then they'll be different. And this is what allows for natural selection and evolution to occur. In bacteria, this is an asexual process, which means it's just one bacteria splits into two. That's it. There's no mom and dad or anything fancy. And these guys are very, very fast and efficient at doing this, at least most bacteria. In this example, we're talking about E. coli. E. coli divides every 10 minutes in ideal conditions. So you have E. coli, and if you let it grow for 24 hours with all the nutrients and space that it can get, unlimited amounts, you would have 4.7 times 10 to the 21 cells, or 47 with 20 zeros after it, which would equal 5,100 tons of E. coli. Literal tons, 2,000 pounds. Does this happen? No. Why? Because there's not enough food. There's not enough space. Okay? So they can't grow like that fast forever. Only until the nutrients run out. Now, for each organism, the amount of time it takes to go from one organism to two is called the doubling time. And the doubling time is intrinsic to the organism, which means each organism has a doubling time that they go by. Okay? And this is based on their genetics. Sometimes you can speed that doubling time up or slow it down a little bit by changing things like temperature or gas or things like that. But for the most part, it is what the organism does. Okay? That is its time that it takes to divide. Average doubling time is 30 to 60 minutes. E. coli has a very short doubling time, 10 to 15 minutes. Some bacteria, though, have really long doubling times. Microbacterium tuberculosis, 36 to 72 hours. Okay, so much, 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 much slower than E. coli. Now, because these organisms are going from one, then into two, and then each of those two new cells, 10 minutes later, can go from one into two again, this is an exponential growth. So if you start off with one, it becomes two. Each one of those two then divides, you get 4. Each one of those 4 then divides, you get 8. And then 16, then 32, then 64, then 128, blah, 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 blah. Okay? So once this gets going, it is very, very, very rapid. Okay? So why are we talking about this? We're talking about this because we can utilize this information to come up with a rough idea of how much organism we have after a certain amount of time. And so now we're going to do a little bit of quick math, everyone's favorite in science. And this quick math is a very simple formula. NF equals NI 2 to the N. NF is the total number of organisms. And this is usually what you're trying to calculate. Okay? And I is the initial number of organisms. So how much should we start off with? How much should we add to our median? And then N is a ratio between the total elapsed time and, or over I should say, the doubling time. And remember, the doubling time is how long it takes one organism to become two. So let's try a simple problem and see how everyone thinks about this formula. So let's say we're looking at E. coli. And as I said, E. coli has a doubling time of 10 minutes. We start off with, let's say, 100 organisms, and we let them go for 10 hours. How many organisms would we have at the end of the day? So what would be the first thing that we want to do if we want to put this into this formula? Do we have all of the bits and pieces we need to put into this formula as is? Do we have Ni? 100, right? Do we have N? Not yet, right? Because N is a ratio. So we have to calculate N first. Okay? So N equals the total elapsed time, which is 10 hours, over the doubling time, which is 10 minutes. In fraction land or science land, is this okay, the way that this is set up? No, because the units are different. So you need to make sure the units are the same. So you either have to convert 10 hours into minutes, or 10 minutes into hours. In this case, it's probably easier to convert 10 hours into minutes. Okay? So how many minutes are there in an hour? 60. And we've got 10 hours. So that means we have 600 minutes. Over 10 minutes. Divide, bing, bang, boom, N equals 60. Now we have everything we need for this formula. Plug it in. NF equals Ni, which is 100, times 2 to the N, which is 60. Done. And the exam, that's all I would expect you to do. Why? Because I don't expect you to bring a calculator, and I also don't expect you to be able to do 2 to the 60th in your head. Okay? Or even on the back of a piece of paper.
I didn't catch what you said 100 was. I know you labeled it as...
Oh, this?
Yeah.
This is the Ni, so the initial number of organisms. So that's how many organisms we stuck into our culture to begin with. Be careful with this. Sometimes people try to oversimplify. This is as far as you can get without doing this exponent. Okay, so remember order of operations? You have to do exponents before multiplication or division. So you cannot try to simplify this and say it's the same as this. It's wrong. Okay? Without doing this exponent, this is as far as you can get. So, if we were to do this equation for multiple different time points, okay, and we plotted it on a chart, this is what it would look like. It would be an exponential growth curve. Okay? It starts off slow, because you literally go from 1, then to 2, then 2 to 4, 2 to 8, but eventually this grows very, very rapidly until you get a very sharp spike. So this is what it would look like in an ideal world. But what did I just tell you before? The world sucks, okay? There's not enough nutrients. There's people fighting you all the time. So what does it really look like? It doesn't look like this. Instead, it looks like this. So if we have time on the bottom, and number of organism side. Everything goes good, fine and dandy. Boom, boom, I'm going exponential. Oh shit, someone starts to take my food. I start to plateau. And then they kill me. Okay? And we can break this up into 4 different curves. The lag phase, the lag curve, the log curve, or exponential as your book calls it. The stationary curve and the death curve. The lag curve, or the first curve, notice that there's not much growth or division. Why? Because this is the point where you just stuck your organism in nutrient material, and instead of dividing, it's getting fatter. Remember I said they can either get fatter or divide. Well, in the beginning, they're just getting fatter. Then they all get too fat, and they say, okay, time to divide. At this point, there's tons of nutrients available, so they're growing at their true doubling time, and this is where it's exponential. Then all of a sudden, they start to run out of nutrients. Either they used it all up or their neighbors did. It's not enough to kill them, because there's still enough around for them to survive, but there's not enough for them to divide anymore. So they plateau, or they stop dividing. Then if no new nutrients are added, all of it starts to be depleted, waste products also build up, and then the organisms start to die because they don't have enough to survive. And we draw it like a dotted line at the end because sometimes some organisms will survive, even in these really nasty conditions. And this is what happens with endospores. So, population growth curve, lag, exponential, stationary, and death. Okay? Here we go, lag, exponential, stationary, and death. Again, lag is where they're getting fatter instead of dividing. Exponential is where they have all the nutrients they need and they're growing and dividing at their true doubling time. Stationary is when they start to run out of nutrients, not enough to kill them, but enough to stop them from growing. And then death is when they run out of nutrients, waste products build up, and they die. This is everything that works. So why the hell do we care about this? Why do we care if we're dealing with pathogens? Well, when we're dealing with pathogens, this becomes important. Because it tells us how many organisms we can expect to find in the body at what point in time. And interestingly enough, the more organism that's there, the more symptoms you get. So when we actually talk about diseases and how they progress in humans later on in the semester, I will draw you an almost identical chart. However, instead of number of organisms on the y-axis, it's going to be the number of symptoms. And that's because the more symptoms you have, the more organism you have, the more symptoms you develop. So if we understand this of the organism, we can figure out how long the disease will be and when will it peak. Okay. Also, this is important if you work in industrial microbiology. Because if you're working in industrial microbiology and you're trying to make a ton of organism, you want to keep them growing right here. That's when you're going to get the most bang for your buck. How do you do that? You put them in something called a chemostat. A chemostat constantly adds in nutrients and takes out waste products so that they always stay in this growth curve. They don't do this. Again, this is not natural. This never happens in the real life or in the real world. It only happens artificially with industrial microbiology. So how can we actually directly measure how many bacteria we have? So what I've just talked about is a hypothetical measurement. But how can I actually take a sample and say, okay, I have this many bacteria in it? Well, the simplest method is to use something called a spectrophotometer. And a spectrophotometer is a machine that has a light bulb in it and a detector on the other side. And what this does is it measures something called turbidity. And turbidity is basically a fancy word for cloudiness of a liquid. So if you have a lot of organism in your liquid, it's going to become cloudy because the organisms take up mass and they block the light. If you have little organism in your material or your media, there's nothing there to block the light, and so now it is translucent and it is not turbid, okay, or less turbid. So with the spectrophotometer, we can take our sample, we can stick it in there, shine light through it, and if all the light goes through it and is measured in the detector, that tells us that it's not turbid, it's clear. There's nothing in there. However, if we have a sample that looks like this and it's got tons of bacteria in it, we stick it in the spectrophotometer, light goes through, but most of it gets blocked, and only a little bit comes through, that tells us that it's turbid as hell. Okay? I won't go any further. I think it turned. I could have said that, but I didn't. So what that means is we can then take the amount of light that comes through from this detector, put it into an equation, and get a rough idea of how many particles are in that material. And if the particles are bacteria, there we go. We just calculated how many we have. Can anyone think of a drawback to this?
Is there a quantitative measurement of this?
So this is relatively quantitative, right? It's relatively quantitative. Because you can test standards. So that's what we would do, is we would take a sample that we know has exactly 5 million bacteria in it, measure it, get a reading, and then we would take our sample, measure it, get a reading, and say, okay, it's that different from this, so that means it's 2 million instead of 5 million, or something like that. The biggest drawback to this is, let's say I took some formaldehyde. Formaldehyde's nasty stuff. It would kill all my bacteria. But my bacteria don't go anywhere. They're still in the material. They still look like this, but they're all dead. So this tells you nothing about if they are alive or not. It just tells you that they're there. And that's not really that important for us, right? We want to know who's actually alive. If they're all dead, I could drink it. Nothing's going to happen. So how do we determine live bacterial counts? Well, one of the simplest ways is to use something called a hemocytometer. This is a special glass slide that has a grid etched onto it, a microscopic grid. We then take our bacterial sample and we mix it with something called trypan blue. This is a blue dye that will stick to dead organism. The live organism doesn't stick to, they stay clear, translucent. So when we squirt this onto the slide, this dye cell mixture, we then put it under the microscope and we count the bacteria that are not dyed blue. That tells us they're alive. We then take that, put it into a formula, and it gives us a rough idea of how many we have. You can do this in about a minute. It's relatively quick. Okay? But with bacteria, it's a little hard because they're really, really small. So it's difficult to see. With animal cells, you do this all the time in tissue culture. And this is how you determine how many you have and what you have to do next. But with bacteria, there are other approaches because they are so small. One of the easiest and cheapest ways is to use something called a Coulter Counter. And this is where you take your cells and you pass them through a single file line, okay, through a electrical detector. And what this detector is measuring is something called resistance. Okay? And resistance is how the electrical field is blocked. Now, if the cell is intact or big, like full, alive, it has more resistance or blocks more electricity. If the cell is dead and broken up, it has less resistance or blocks less electricity. So you can pass your cells single-filely, and you say, oh, this one's got a lot of resistance, that counts as one live cell. Oh, this one doesn't, that counts as one dead cell. And you do that really, really fast with millions of cells, and that gives you a count of how many are alive. Sounds cool, but it's really bad. It doesn't work very well. Okay? It was good in its day in the 70s when they developed it, but it's not really used anymore. So instead, what most people use now is something called a flow cytometer. This is virtually the same premise, single-file cells passing through a liquid. However, instead of using an electric field, you are using lasers. These lasers excite the cell or light it up, and then they can be detected using a detector. And so you can tell the size of the cell, what's inside of it, or the granularity. And you can even add things onto the cell that glow or fluoresce, and so you can even tell what's on the surface of your cell, what's inside of your cell, a whole bunch of different stuff. Most fancy flow cytometers anymore can detect up to 12 to 18 colors. So you can look at 12 different things on one cell at one time. However, these machines are very expensive. A couple $1 million on average. It's not something we have here. We have a lot of them at Case, though. I use these guys all the time. Another cheaper route that you could use is something called real-time PCR. PCR, polymerase chain reaction, makes copies of DNA. So what you could do is you could take your cells, pull out their DNA, and then make copies of it. And you can say, okay, if every cell just has one piece of DNA or one chromosome, and I make copies of it and I end up with 5,000 copies, that tells me I started off with, let's say 500 organisms. okay? Because each cell has one chromosome. The drawback to this approach is sometimes cells divide. And when they divide, they now have 2 chromosomes. So with this approach, you can be off by a factor of 2. Okay, if all of your cells are dividing at once, it looks like you have a hell of a lot more than you actually do. And this is just an example of a Coulter counter. So cells pass through single file and electrical detector, which measures the resistance between the cells. And that's it for chapter 7. Any questions? Other than it's super boring. Cool.